Have you ever come across something that just clicks? Like it unlocks a totally new angle on a really tough problem?
Mm-hmm.
Well, today, we're diving headfirst into some research that feels a bit like that. It's possibly hinting at a shortcut for understanding, maybe even tackling, some really challenging neurodegenerative diseases.
Yeah, it's a fascinating study. We're looking at research out of Northwestern University, actually just published last month, March 3rd, 2025, in PNAS.
Right.
And it centers on this experimental drug, NU9. Now, it's already under investigation for ALS.
Okay.
But the real kicker, the thing that's really grabbed attention, is its unexpected potential for Alzheimer's disease, too.
Exactly. ALS and Alzheimer's, I mean, on the surface, they seem quite different, don't they? Different battles, different symptoms mostly.
Absolutely.
Yet this research suggests maybe there's a shared vulnerability. So for everyone listening, our mission here in this deep dive is to unpack what this study found and really get why it's causing such a buzz. We'll try to do it without getting bogged down in super technical jargon.
Right, focusing on that core idea, the possibility of a common mechanism linking these diseases.
Yeah. So where do we start? What's the common thread?
Okay, well, let's look at that common ground. Both ALS and Alzheimer's fundamentally involve this harmful buildup. Proteins that should be doing their job correctly, they misfold.
They go wrong somehow.
Exactly. Think of it like, I don't know, parts in a complex machine being made incorrectly. So in ALS, a major player is the misfolded SOD1 protein.
Okay.
Whereas in Alzheimer's, we're mainly talking about those sticky clumps of amyloid beta oligomers.
So these proteins that are normally fine, they basically go rogue. I think one of the researchers used that term.
Yeah, William Klein described them that way. They form these toxic aggregates, these clumps in the brain.
And that's what messes things up.
That's the thinking. This clumping disrupts how brain cells function normally and eventually leads to neuron death. It happens in both diseases, even though the specific protein causing the trouble is different.
Right. Different culprits, same kind of downstream damage.
Pretty much.
So how does NU9 fit in? What makes its approach different?
Well, this is what's so interesting. NU9 doesn't seem to directly attack those specific misfolded proteins, not SOD1 in ALS, not amyloid beta in Alzheimer's, not directly.
Right. Oh.
Instead, it seems to be working on a more, let's say, fundamental level. It appears to influence some underlying cellular process that's involved in this neurodegeneration.
Ah, okay. So instead of just, like, mopping up the mess, the symptoms, maybe NU9 is addressing a root cause, like a shared weakness in how the cells handle these bad proteins.
That's the idea. And Richard Silverman, who actually developed NU9, he highlighted this. He called it quite remarkable, that the drug shows activity in these multiple systems.
And that's the core of the excitement, isn't it? For listeners, I mean. A single compound showing potential across different, really devastating diseases.
Absolutely. That's the potential breakthrough here.
So what did the study actually find? How does it seem to work?
Okay, so the Northwestern study gives us some initial clues. They started with neuron cultures in the lab.
Right.
And they found that NU9 did reduce the buildup of that amyloid beta. But what's really noteworthy, I think, is that this protective effect seemed to hang around.
Hang around how?
Well, it persisted even after they removed the drug from the culture.
Oh, wow. Okay, so it's not just like a temporary fix, a Band-Aid while the drug is present.
Doesn't seem like it. It hints at maybe a more lasting change to the cell's machinery.
Okay. And did they test this in animals too?
They did. They moved to a mouse model of Alzheimer's disease. And again, encouraging signs. The mice that got NU9 orally actually performed better on memory tests.
Better memory scores. That's significant.
It is. And there was another finding, too, related to inflammation.
Which is a big factor in Alzheimer's, right? Brain inflammation.
A very big factor. And the study showed NU9 reduced that inflammation in the Alzheimer's mouse models.
So it's hitting multiple things, memory, inflammation.
Seems like it. William Klein, who we mentioned, he really emphasized this broad action. He said the drug showed significant power at both the cellular and whole animal levels.
Okay, cellular and whole animal. So let's drill down on the cellular part. How? What's the mechanism they think is at play?
The evidence points towards lysosomes.
Lysosomes.
Yeah.
Okay, refresh my memory. Those are like the cells.
Recycling centers, yeah, or waste disposal units. They break down unwanted material.
Got it.
And inside the lysosomes, there's a key enzyme called cathepsin B. Now, in Alzheimer's, it looks like this whole cleanup system gets overwhelmed or impaired.
So the trash piles up, the amyloid beta.
Essentially, yes. And the hypothesis is that NU9 might somehow help shuttle those amyloid beta proteins into the lysosomes
Okay.
so that cathepsin B can then do its job and break them down.
Ah, so NU9 is like helping the garbage collectors get the trash to the incinerator more effectively.
That's a pretty good analogy, yeah. Helping the cell's waste management system get back on track.
But it sounds like maybe they don't know exactly how it's helping the garbage collectors.
Precisely. That's the catch right now. Klein called the exact target of NU9 within the cell a bit of a black box phenomenon.
Black box, okay.
They know it's triggering this lysosome-related process involving cathepsin B, but they haven't pinpointed the exact molecule or switch that NU9 flips first.
I see.
They use another analogy like a relay race for these toxic proteins inside the cell, and they think NU9 might be intervening early in that race, somewhere before the proteins actually get handed off to the lysosome for disposal.
Okay, so for us listening, the key point is we have a good idea of the result.
Yeah.
NU9 seems to boost the cell's cleanup crew, but the exact first domino it pushes is still under investigation.
So that's a good summary, yes.
But even with that bit of mystery, the implications here feel, well, pretty huge.
Oh, absolutely. I think the most significant takeaway really is this potential for a shared fundamental mechanism across different neurodegenerative diseases.
Right.
Silverman himself said their findings indicate that common mechanisms might connect them, and that, he suggested, could pave the way for a whole new family of therapeutic compounds.
A new family of drugs.
Wow.
That could target multiple diseases.
That's the potential.
That really shifts the paradigm, doesn't it? Instead of one drug for ALS, another completely different one for Alzheimer's, maybe another for Parkinson's.
Exactly. We might be able to target a more fundamental cellular weakness and potentially intervene earlier in the disease process, maybe before massive cell damage occurs.
So what's next for NU9 then? More research, I assume.
Definitely. Klein mentioned needing more rigorous, more in-depth memory testing in the animal models.
Makes sense.
And Silverman's team at Akava Therapeutics, the company developing NU9, they're working on refining the compound itself, maybe making it even better.
And are they looking beyond ALS and Alzheimer's?
Yes. There are plans to explore if NU9 shows similar benefits in other conditions, like Parkinson's disease and Huntington's disease.
Interesting. It's probably worth mentioning who these key researchers are, just briefly.
Good idea. Richard Silverman, as we said, developed NU9. He's also perhaps famously the inventor of the drug Lyrica.
Oh, right. Lyrica, okay.
Yeah, so a seasoned drug developer. And he founded Akava Therapeutics. William Klein is a very well-respected Alzheimer's researcher, co-founder of another company, Acumen Pharmaceuticals, also focused on Alzheimer's treatments.
And they're both at Northwestern.
Both affiliated with Northwestern University, yes.
Yeah.
And the funding came from places like the NIH and Northwestern's Chemistry of Life Processes Institute.
Crucial support for this kind of work.
Indeed. And one more piece of context, NU9 already has the green light from the FDA for clinical trials in ALS patients.
Ah, okay. So it's already being tested in humans for ALS.
Yes, which gives us some initial data on its safety in people, which is always a critical step.
Right, absolutely. Okay, so let's try and summarize this deep dive for everyone listening. What's the big picture?
The big picture is we have this experimental drug, NU9, started with ALS in mind, now showing really promising, though early, results in models of Alzheimer's disease, too.
And the key seems to be that it's not targeting disease-specific symptoms directly, but rather a more fundamental mechanism.
Involved in how cells handle these misfolded proteins, likely involving the lysosomal system.
Yeah. It's definitely early days, we should stress that.
Oh, absolutely. Early days.
But the prospect, the idea of a more universal treatment strategy for these truly devastating diseases, it's incredibly exciting.
It really is. This research kind of nudges us to think differently, doesn't it?
How so?
Well, instead of seeing ALS, Alzheimer's, maybe Parkinson's as totally separate problems, it highlights these potential shared vulnerabilities deep down at the cellular level.
Vulnerabilities that maybe we can target.
That's the hope, target them therapeutically.
And that really does make you wonder, doesn't it? If diseases that look so different on the surface, like ALS and Alzheimer's, could share these deep biological connections, well, what other conditions that we think are totally unrelated might actually be linked at some core level?
It's a powerful question.
Yeah. Sort of pushes you to think about the broader implications, the unexpected connections that science might still uncover about how our bodies work and how they sometimes go wrong. Lots to think about there.